Some children begin seizing before they ever leave the hospital, in the first days of life, before a face is fully recognized, before a hand can grip. A seizure. One missing copy of one gene is enough. Its name is STXBP1. STXBP1 sits on chromosome 9. It builds a protein called MUNC18-1. And MUNC18-1 has one job at every synapse in the brain, readying the tiny sacs of neurotransmitter for release. Those sacs, synaptic vesicles, line up at the edge of the neuron like runners on a block. MUNC18-1 is what puts them there.
But not every synapse sends the same message. Some are excitatory, the accelerator, pushing the next cell to fire. Others are inhibitory, the brake, holding it back. A healthy brain holds the two in balance. Weaken the brake, and excitation runs away, and that runaway wave of firing is a seizure. For years, children with this condition had no diagnosis. Then, in 2008, Hirotomo Saitsu, Mitsuhiro Kato, Takeshi Mizuguchi, and their colleagues at Yokohama City University found the culprit. In 5 children with catastrophic early seizures, the same gene was disrupted. In one, a deletion had swept it away. In the others, a single letter change had broken it. And every change was new in the child, carried by neither parent. Published in Nature Genetics, one of the first genes ever tied to this earliest, most severe epilepsy.
Most of the harmful variants simply stop the protein mid-build, a truncated MUNC1 that the cell discards. So the neuron runs on half the normal supply. Half is not enough. And a subset of missense variants go further. The altered protein actively traps other key release factors, pulling them away from the synapse. Either way, the machinery is short. To release its cargo, a vesicle must fuse with the cell wall. That fusion is driven by snare proteins, syntaxin and SNAP25 waiting on the wall, synaptobrevin hanging from the vesicle. MUNC18-1 is the starter. First, it clamps syntaxin shut, folded on itself. Then, on cue, it opens syntaxin and lines it up with SNAP25. Synaptobrevin drops in from the vesicle, and the 4 strands zip together, top to bottom, into one tight bundle, hauling the 2 membranes together until they fuse and the cargo spills out. Its work done, MUNC18-1 is pushed aside. Without it, syntaxin never opens, the bundle never forms, the vesicle docks but stays silent.
Now have the MUNC18-18. The vesicles are there, the signal arrives, but the priming step is sluggish. And the neurons that suffer most are the inhibitory neurons, the cells whose job is to calm a circuit down. Mouse models built to mimic this, cutting MUNC18-18 to half in only the inhibitory cells, develop epilepsy, movement problems, and cognitive deficits, just as the children do. Have the silence, and the circuit tips into a storm. Intellectual disability touches every child with this condition. Seizures follow most, often in the first weeks of life, before any treatment plan is in place. Movement disorders come too. Tremor, unsteady gait, repetitive movements that are not seizures but look like them. And in adults, 8 in 10 still have active seizures, most managed on several medicines at once. There is no approved treatment that changes the course, but the logic points straight at what to try. If the problem is too little protein, could you make the healthy copy do more? That is the idea behind an antisense oligonucleotide, a short synthetic strand matched to one stretch of the gene's own RNA message. It clamps onto a control site where a silencing molecule, a microRNA, normally docks to quiet the message. Take that seat, and the message is read more, lifting MUNC18-1 toward a full dose. Gene therapy vectors that deliver a second working copy are in development too. It is early work, nothing is approved, but the target is clear and the path is lit.
18 years, from 5 children whose seizures had no name, to a mechanism we can read, to the first attempts to restore what is missing. The families made that happen. They are still moving it forward. STXBP1 Foundation, built by parents, funding the search. Found in a lab, carried by families. Find them at stxbp1disorders.org. The Gene Channel.